Assalamu alaikum. Dear students, I'm here Hafiz Muhammad Ammar, co-teacher for microbiology course code BOTN 4128. In continuation of previous lectures, today we are here for lecture number 16 entitled Chemical Requirements of Microbial Growth, including phosphorus and other requirements. The contents which will be covered today's lecture include sulfur, phosphorus, trace elements, organic material, nutrient as limiting factor, and nutrients supply. Sulfur, it is the most common element by mass in the universe, ranking as 10th most common element and 5th most common element on earth. Historically and in literature, sulfur is also called burning stone. Hydrogen sulfide gives the characteristic odor to rotting eggs and other biological processes. Sulfur is an essential element for all life, but almost always in the form of organosulfur compounds or metal sulfides. Sulfur is one of the core chemical elements needed for biological functioning and is an elemental macronutrient for all living organisms. In addition to carbon and nitrogen, microorganisms need a supply of certain minerals, especially sulfur and phosphorus, which are very important cell components. Microorganisms obtain sulfur from inorganic sulfate salts and from sulfur-containing amino acids. 3 amino acids and 2 vitamins, including biotin and thiamine, are organosulfur compounds. Many cofactors are also containing sulfur, including glutathione, thioredoxin, and iron sulfur proteins. Disulfides confer mechanical strength and insolubility of the protein keratin, which is found in other outer skin, hair, and feathers. These disulfides are as sulfur-sulfur bonds. They use sulfur and sulfur-containing amino acids to make proteins, coenzymes, and other cell components. Some organisms can synthesize sulfur-containing amino acids from inorganic sulfur and other amino acids. Role of the sulfur in microbial growth. The inorganic sulfur compounds of biological relevance, which occur in the biological sulfur cycle, are elemental, like sulfur, sulfate, sulfite, thiosulfate, polythionates, sulfide, and polysulfides, as shown in the figure. Sulfur can adopt many oxidation states, ranging from 2 to 6. Inorganic sulfur compounds of intermediate oxidation states can serve as electron acceptors or donors in redox process. In contrast, sulfate and sulfide cannot be further oxidized or reduced. They are therefore the final products of most sulfur oxidation or reduction pathways. The biological roles of inorganic sulfur compounds are rather restricted. Either they are served as acceptors or donors of electrons for dissimilatory energy generation electron transport, which is almost exclusively among prokaryotes. or they are employed as sources for sulfur assimilation. It is very common in prokaryotes as well as in algae, fungi and plants. In this picture, solid lines indicate aerobic reactions, while dashed lines indicate anaerobic reactions, and dotted lines indicate both aerobic and anaerobic activity. Desulfurization by many aerobic and anaerobic prokaryotes, assimilatory sulfate reduction by many aerobic and anaerobic microorganisms, and disproportionation of thiosulfate by desulfovibrio and desulfocapsa bacteria. In state 1 and 2, sulfide and sulfur oxidation by colorless sulfur bacteria as shown in the figure. Number 3 is the sulfur reduction by the anaerobic microorganisms. Number 4 and 5, these are the anaerobic sulfide and sulfur oxidation by purple sulfur bacteria and green sulfur bacteria, while 6th is the sulfite reducing bacteria's reaction. Phosphorus. It has a concentration in the Earth's crust of about 1 gram per kg. Phosphorus is a macronutrient necessary to all living cells. It is an important component of adenosine triphosphate, nucleic acid, maybe DNA or RNA, and phospholipids in cell membrane. It may be stored in intracellular volutin granules as polyphosphates in both prokaryotes and eukaryotes. It is a limiting nutrient for algal growth in lakes. The average concentration of total phosphorus, organic phosphorus and inorganic phosphorus in wastewater is in the range from 10 to 20 milligram per liter. It is very high. The phosphorus cycle. It is the biochemical cycle that describes the movement of phosphorus through the lithosphere, hydrosphere and biosphere. Unlike many other biogeochemical cycles, the atmosphere does not play a significant role in the movement of phosphorus, because phosphorus and phosphorus-based compounds are usually solids at the typical range of temperature and pressure found on the earth. So phosphorus in agriculture soils originates primarily from two sources: the parent material from which the soil developed, or phosphorus containing amendments applied by producers. Parent material soil phosphorus originates from the weathering of primary minerals such as apatite. Phosphorus from plant residues and animal wastes is primarily in the organic form, and so must be mineralized before it can be taken up by plants. It is a biologically controlled process that depends on the carbon phosphorus ratio. Leaching is the removal of dissolved phosphorus from soil by vertical water movement. Runoff is the removal of soil bound and dissolved phosphorus by overland flow. Adsorption is the chemical binding of plant available phosphorus to soil particles, which makes it available to plants. Desorption is the release of adsorbed phosphorus from its bound state into soil solution. Absorption is the diffusion into interior spaces and assimilation within the soil particles themselves. Sorption is the general term used when the specific removal process is not known, and the precipitation reaction can occur concurrently and so are often included within the sorption term. Microorganisms obtain phosphorus mainly from inorganic phosphate ions. They use phosphorus as phosphate to synthesize ATP, phospholipids, and nucleic acids. Phosphates from fertilizers, sewage, and detergents can cause pollution in lakes and streams. Eutrophication. Eutrophication is an enrichment of water by nutrient that leads to structural changes to the aquatic ecosystem, such as algae bloom, deoxygenation, and reduction of fish species, etc. The primary source that contributes to the eutrophication is considered as nitrogen and phosphorus. When these two elements exceed the capacity of water body, eutrophication occurs. Phosphorus that enters lakes will accumulate in the sediments and biosphere. It is also can be recycled from the sediments and the water system. Over-enrichment of phosphate in both fresh and inshore marine waters can lead to massive algae blooms, which when they die and decay leads to eutrophication of fresh waters only. You can see in the pictures. An example of this Canadian experiment lake area shown in picture. These freshwater algae blooms should not be confused with those in saltwater environments. Recent research suggests that a predominant pollutants responsible for algae blooms in saltwater and coastal marine habitat, that is nitrogen. Mainly inorganic phosphate salts and buffers are the main source of phosphorus. Arbuscular mycorrhizal fungi. On the land, phosphorus gradually becomes less available to plants over thousands of years since it is slowly lost in runoff. Low concentration of phosphorus in soils reduces plant growth and slows soil's microbial growth, as shown in the studies of soil microbial biomass. Soil microorganisms act as both sinks and sources of available phosphorus in the biogeochemical cycle. Locally, transformations of phosphorus are chemical, biological, and microbiological. Mycorrhizal fungi are described as the crucial microbial group in soil systems, presenting mutualistic association with plants. The occurrence of this group is considered one of the most important microbial indicators of soil quality. The symbiotic association known as mycorrhiza improves plant nutrient uptake and water absorption. It reduces the effect of environmental stress on plants and also protects plants against pathogens. Arbuscular mycorrhizal fungi can increase phosphorus uptake and plant diversity. This suggests AMF may be a major contributor to plant productivity and variability. Phosphate solubilizing bacteria. These are beneficial bacteria capable of solubilizing inorganic phosphorus from insoluble compounds. Phosphorus solubilization ability of rhizosphere microorganisms is considered to be one of the most important traits associated with the plant phosphate nutrition. It is generally accepted that the mechanism of mineral phosphate solubilization by PSB strain is associated with the release of low molecular weight organic acids, through which their hydroxyl and carboxyl groups chelate and cations bound to phosphate, thereby converting it into soluble forms. Phosphate solubilizing bacteria have been introduced to the agricultural community as phosphate biofertilizer. As phosphorus is one of the major essential micronutrients for plants and is applied to soil in the form of phosphate fertilizers. However, a large portion of soluble inorganic phosphate which is applied to soil as chemical fertilizer is immobilized and become unavailable to plants. Currently, the main purpose in managing soil phosphorus is to optimize crop production and minimize phosphorus loss from soils. Phosphorus solubilizing bacteria have attracted the attention of agriculturists as soil inoculums to improve the plant growth and yield. When phosphorus solubilizing bacteria is used with rock phosphate, it can save about 50% of the crop requirements of phosphatic fertilizer. The use of phosphorus sulfur bacteria as inoculum increases phosphorus uptake by plants. Trace elements. These are metal ions required by certain cells in such a small amount that it is difficult to detect. And it is not necessary to add them to culture media as nutrient. These elements are required in such a small amount that they are present as contaminants of the water or other media components. As metal ions, the trace elements usually act as cofactor for essential enzymatic reactions in the cell. One organism's trace element may be another's required element, but the usual cations that qualify as trace elements in the bacterial nutrition are manganese, cobalt, zinc, copper, iron, and molybdenum. Many microorganisms require a variety of trace elements. Tiny amounts of minerals such as copper, iron, zinc, and cobalt, usually in the form of ions. Trace elements often serve as cofactor in enzymatic reactions. All organisms require some sodium, chloride, and halophiles require large amount of these ions. Potassium, zinc, magnesium, and manganese are used to activate certain enzymes. Cobalt is required by organisms that can synthesize vitamin B12. Iron is required for the synthesis of heme-containing compounds such as the cytochromes of the electron transport system and for certain enzymes. Although little iron is required, a shortage severely retards growth. Calcium is required by gram-positive bacteria for synthesis of cell walls and the spore-forming organisms for synthesis of spores. Many microorganisms are unable to synthesize certain organic compounds necessary for growth, and they must therefore be provided with them in their growth medium. These are termed growth factors, of which three main groups can be identified: amino acids, purines and pyrimidines, and vitamins. Vitamins are complex organic compounds required in very small amounts for the cell's normal functioning. Microorganisms vary greatly in their vitamin requirements. Many bacteria are completely self-sufficient, while protozoans, for example, generally need to be supplied with a wide range of these dietary supplements. A vitamin requirement may be absolute or partial. An organism may be able, for example, to synthesize enough of vitamin to survive, but grow more vigorously if an additional supply is made available. In regard to required nutrients for proper growth, there are often limiting factors involved, may be called limiting nutrient, that affects and controls the growth. The availability of specific nutrients dictates organismal growth by controlling and limiting activation of cellular and metabolic pathways which are necessary for the progress. When all nutrients and parameters are ideal and constant during the growth phase, that is regarded as the steady state, in which all requirements are present and microorganisms are surviving. In circumstances where there are less than ideal parameters, such as the lack of specific environment requirements, the growth process is affected. In industrial microbiology, this concept is critical as microbial growth and production is dictated by proper cellular growth and metabolism. The production of necessary components is often controlled by the presence and concentration of a limiting nutrient. Hence, it is critical to identify the required nutrients and ensure these are supplied in the culturing microorganism. Crop residues is a major source of energy and nutrients for microbial growth. Can be converted into soil organic matter. Here is shown an experiment which was conducted to study the supplementary nutrients effect on growth. Nutrient supply in wheat enhanced the wheat residue carbon mineralization, microbial growth and microbial carbon use efficiency when residues were supplied at high rate in the contrasting soil. The results indicate a higher efficacy of soil organic matter formation from returning to residue soil under. The findings of the integrated residue nutrient management on dynamics of residues derived microbial biomass, carbon and residues carbon use efficiency can be used to improve predictive performances of the models on specific soil functions such as soil organic matter storage in agroecosystems. These are the books which were studied for the preparation of this lecture. You can study and available for reference. I hope you understand well the role of micronutrients, trace elements, organic matters in the growth. Thank you very much.